Yeah, thank you very much, first of all, and all of you. Happy Teachers' Day. And I think I misfit in this particular symposium, what has been conducted, and I am following it since yesterday. And I really enjoyed some of the talk. And you could see that you are talking about the data management and genomics and bioinformatics. And I thought that why I should have talked. This is something I was thinking that, okay, I should have chosen a topic which would be in the similar field where you all are talking about the data management, bioinformatics, genomics. But somehow I don't know why I picked the area which is little different, which is not very dry, and which would also bring a different perspective for all of you, that what you could think of using the data, the genomics or the bioinformatics for the advancement of the technology. And this is where I thought, let me talk something different. So liquid biopsy is something I think most of you know what is biopsy. So biopsy is something where you take the tissue, and we call it is an invasive process because you have to go through the surgery, either small surgery or bigger surgery. So the problem is that, yes, it is expensive. It actually exposes you to many more things. So why not opt for a method which would give you similar answer what a biopsy could give you? And this is what the term has came, liquid biopsy. So liquid biopsy is anything which would be minimal invasive or non-invasive way. You get the specimen and you figure it out, the similar kind of attributions what you could get. So this is, you could see the application. When we talk about the liquid biopsy, you can actually do real-time monitoring of the disease or the therapy. You can check the disease relapse. You can actually pick the therapeutic targets. You can stratify the therapeutic intervention. And the best use of liquid biopsy would be screening or early detection of cancer. So liquid biopsy is anything which I mentioned you would get in a non-invasive way. So you use your urine. If you get the urine sample, and if you ask the similar kind of questions that, okay, if a person having the cancer, mostly in bladder or prostate cancer, you could ask that, okay, if the tumor cells are there, and if you take those tumor cells and you can figure it out about the genomics, you can do proteomics or metabolomics. It could be central nervous system where you take the CSF. It could be head and neck, we call saliva. So saliva is another very good example that you could take the saliva and again do such kind of downstream processing. And what could be the biomolecule you'd obtain in the liquid biopsy material? It could be exosome, it could be circulating tumor DNA, it could be circulating tumor cells. And this is where you could see, if you take blood, it has two components. The first component, the upper component, and the lower component. Now the upper component, we call it plasma. And what you expect in the plasma is the DNA. So very simply, you can isolate the DNA. Once you have the DNA, you could use this DNA for doing different kind of genomics. And the genomics, what it will give you? It will give you that if a gene has been amplified, if there is a translocation, mutation, or any chromosomal abnormalities. Now, the very interesting part is the lower part, where the cells are there. The cells are sedimented. And what these cells are, we call it tumor cells. This is called circulating tumor cells. The cells present in the blood of a cancer patient. And what you can use eventually these cells for? Of course, genomics. You can do genomics, but in addition, what you could do, you can use these cells for doing proteomics, because you are getting the whole intact cells. And you can culture these cells. You can do multiple downstream experiments to ask the questions that how the tumor has been initiated, if it has a metastatic property. So the picture, what it gives you, that the liquid biopsy is something which is giving you a clarity about the tumor progression. If you get the cells, if you get the DNA, you could ask multiple questions. So the area has been separated. If you see the last decade, I would say, you keep hearing in any clinical setup that we are talking about the circulating tumor cells, we are talking about the circulating tumor DNA. So the very basic questions comes in mind that why this is important. And as I mentioned earlier, that it would be a non-invasive sampling. You could go to patient and ask 10 times blood sample, but you may not ask a patient to go for surgery 10 times. So this is a big boon in the area of cancer research. Localized cancer, again, early detection is the key. And the biological insight, that is where we call, you could understand that what is something happening in the tumor. So if you talk to any cancer biologists, they will ask you that, or you would ask a clinician that what is something which bothers you maximum. And me as a clinician or me as a researcher, I would say that the three biggest challenges what we face in cancer is first metastasis, because we do not know that the tumor cells reach to what are the different distant organ. The second thing is that the patient has been given to different kind of therapy. It will either chemotherapy, targeted therapy. But what happened eventually over the time, the patient become resistant to the therapy. And this is where we called it therapy resistance. The third biggest challenge is the tumor relapse. The patient responded to the therapy, and later the clinician realized that after a couple of years or 10 years, the tumor comes back. And this is where we call tumor relapse. And in this field, we definitely would like to find a way where we can address all three biggest challenges. So the way we understand the cancer, how we detect, the very basic way is that you do a scan. So the first thing you present in the clinic, and the clinician will ask you that, can you just go and do the scanning? The second level where we talk about the molecular pathology. So we believe in the pathology and we ask you to do, okay, can you do immunohistochemistry to see the cellular population? However, the NGS had made a big revolution in the field. And it is not in cancer, it is in every diseased area where NGS contributed immensely. And this is where NGS has given us tons of data. So we were talking about the terabyte, petabyte. So you can see the data we have collected, and our understanding about the disease has enormously increased, the depth we understood. So the NGS we are using very routinely. And what NGS give you, that you can do whole exome sequencing, whole genome sequencing, you can find such kind of mutations. And on that particular mutation, if there is a targeted therapy existing, it is something like a miracle that the patient has been given to that particular therapy and patient responds very well. But what has changed, I would say, for last 5 years, the liquid biopsy came. And many of the clinics, we are practicing actually the liquid biopsy. How? Because what I mentioned you that it could monitor the cancer in blood. This is something very interesting, what I'm working, my group is working. So we are working on both the aspect of CTCs, circulating tumor cells, and ctDNA. CTCs, because I mentioned you, it is coming from the tumor present in the blood. So if you can isolate these CTCs, what you can do, you can extract the DNA from the CTCs. If you can extract the DNA from the CTC, you can do whole genome sequencing, whole exome sequencing. You could identify the predicted, you can predict the mutations, identify the potential mutation, and you could go next stage where you can do the targeted genomics. So this is the one aspect, and it could be used for any kind of cancer. The other aspect is that you can directly take the ctDNA from the blood. You may not need to take the cells, but you can take the plasma part. Again, ask the similar question. You do genomics, find the mutation, and target those mutations. But what is interesting is that the second part, you see that the genomics would be the one of the approach. But why not if you can isolate those circulating tumor cells and culture them? The culture of these tumor cells in ex vivo condition would again give you another platform. It would actually maneuver an experimental biologist or a clinician to understand that how the tumor cells proliferate, and if it shows any different kind of phenotype. This is what we have done. We have established the method where we can culture these cells, and what you could see that if you can culture these cells, your goal would be that you can do ex vivo way the drug susceptibility testing. You can treat different kind of drug combination to these tumor cells in the plate. And this is what we call the personalized therapy, that for every patient, you know that which drug combination would be effective. So this is something we are working on. So I'm going to focus now mostly on the circulating tumor cells. What are these cells? These are the cells which are coming from the primary tumor. Intravasate in the blood, stays in the blood, extravasate at particular time, involves in metastasis. And this is something, the edge, what CTCs are giving to the clinician is that the size of the tumor, if it is very small, you can imagine the size 0.05 millimeter. You can imagine how small it is. We cannot detect the cancer. We don't have any technology, any method right now. Once the size becomes 10 times bigger, 0.5 millimeter, you could only detect using circulating tumor cells. However, when we use the imaging, the size of the tumor is already 100 times bigger. It's a 5 millimeter. And most of the time we wait for the clinical symptoms. And we wait, once we wait for the clinical symptom to appear, you could imagine the size, and the cells are already in the metastasis. Tumor cells are already reaching to the distant organ. So this is the biggest advantage working in this area, liquid biopsy. Why the world is talking about this, and why a lot of effort has been done. And this is giving you a clear picture that how many exploration has been done on different kind of cancer. And of course, prostate, breast, rectal, and lung are the top ones, where many more research papers have been published. There are a lot of research groups who are working on it. And this is how you could see that there are efforts to characterization of the CTCs, tumor cells. People are doing genomics, people are doing proteomics. So all such kind of dimensions people are exploring. So this is what we use to isolate the circulating tumor cells. This is based on the two physical property of the tumor cells. One is what we call it the plasticity, and the second is the size. So you could see that this is a spiral macrofluidics we use, where the blood is inserted and the blood is going in the spiral macrofluidics, and there are two outlets, outer outlets, inner outlets. So what you would see that in the inner outlets, the cells coming, these are the circulating tumor cells. The outer outlets, you are getting WBCs. And this is how this looks like. The tumor cells going through the capillary with other blood cells, and it is getting collected actually to the inner wall and the outer wall. So the method has been exploited. We established a method where we can culture these circulating tumor cells. And this is how you can see that the tumor cells in the culture, how they look like. A single CTCs and CTCs in clusters. So these are the tumor cells. And what you see that a tumor cell has a property. It has big nuclei. So this is the very simple way a pathologist or a cytopathologist will check under the microscope they should actually do the expression of cytokeratins. And what are these cytokeratins? These are the intermediate filaments. You know intermediate filaments. Every epithelial cell should have. So this is what you can see, that green, what you see, these are the cytokeratins. And blue, what you see there is the nucleus, which tells that these are the tumor cells growing in the culture method. I'm not expending the technique, but the goal is to make you understand that how you can explore the method, how you can use the blood sample to culture these circulating tumor cells. And this is how in culture you could see the tumor cells over the time, it will expand. And there's expansion. If you get the tumor cell, 5 tumor cells at day 0, over the time 5 become 10, 10 become 20, 20 become 50. So this is how it grows exponentially. And this is where even you would able to achieve less number of cells. If you can establish a method where you can culture these cells, you can actually get good number of these cells present in the microvales. This is showing that how these tumor cells clusters look like, and what actually you could see that it's the effect of hypoxia, the way we grow these cells is in hypoxic condition, and hypoxic condition makes the best out of the culture. And this is what you could see that when we cultured them for day till 3 weeks time, day 14 you can see that it gives the much more intact cluster. And this is something again, the culture, CTC cultures, which are growing on day 14, and it has been stained for cytokeratin. Bright field image, you could see that it looks like the tumor cells are getting clustered. DAPI is the nucleus, blue is the nucleus, and CD45, which you see, there's nothing you can see in the picture, which is good. This is how it should have negative for the leukocyte. It shows that I'm not growing any other cells, I'm only growing the tumor cells. That's why it is showing CD45 negative. This is what you could see, what we have done. We have cultured the cells, and again different kind of markers, pan-cytokeratin, vimentin, and these are the merged figure. And interestingly, what you could see, this is actually the 3D culture. So what I was discussing so far earlier was 2D culture. Now the lower panel, it shows the 3D culture, and you could see that the tumor cells are making different kind of phenotype, epithelial phenotype, mesenchymal phenotype. There are some population where epithelials are more than the mesenchymals. There are some population mesenchymals are more than epithelial. So there are 5 different kind of phenotype you would see when you grow these tumor cells. And this is something which had been shown earlier also from the group from Harvard. It was published in Science. And what we have seen similar kind of results when we were growing these cells in the 3D culture. So this is very interesting. It shows that how the tumor cell behave. And this is how it look like. A tumor cells which comes in the blood, it can go in different kind of phenotype, either epithelial or mesenchymal or the intermediate phenotype. Once it acquired this phenotype, it actually is extravasate from the blood vessel and reaching to the distant organ for the metastasis. And this is where you could see the liver, the cells are reaching, it is colonizing, and again ready for proliferation. And this is where you can MET, that the mesenchymal cells become epithelial cells for the colonization. And what you would see that there's an early relapse when these tumor cells are sitting in some of the organ for years and years, and then suddenly they started dividing because there's a MET. And there's a switching of the transcription factor, which again start the cascade of cellular activation. And this is where the cells again goes in the blood vessels. So the first part which would give you an idea about that, how the tumor cells has been characterized, and what we have realized that it actually adhere the intermediate phenotype in terms of epithelial or mesenchymal classification. And the part of the region is that once it is staying in the intermediate state, it has highest plasticity. It can choose to go back or in forth, both ways. And for the cells to come back to the epithelial state, it makes sense because the entropy would be less for cells to go back and start dividing from the intermediate stage. So I would now little bit talking about the clinical aspect. What I have shown you, it was actually the experimentalist, as experimentalist, how we think, how we grow the cells, what are the questions we ask in the lab. But does it make sense to a clinician? If I am going to a clinic and asking a clinician that I am growing the tumor cells, are you interested? For them to put his or her interest in my experimental plan, there should be a translation from bench to bed. And this is what you could see, that when we were culturing these cells, what we could see that they are making different kind of clusters on day 14. And what you should see that when there is a tumor cells present, it makes a multi-layer cluster, which is on the top. When you take the blood from the healthy individual, it will only make cell debris. So this is amazing readout, because you can just seeing these clusters, you could tell that if the tumor cells are present or not. So this is where we have published this work. You could see that you take these clusters from the blood, you culture them, and you look two different kind of phenotype. One with the clusters phenotype, one where there is no tumor cells and it only makes cell debris. This is how the CTC looks like. I showed you earlier. The cells had been stained with different kind of markers. The cells are coming in the clusters. And this is how the clusters look like, which has the staining for cytokeratin, staining for the nuclei, and at the same time, they are negative for CD45. This is the interesting data. We talk about the heterogeneity across tumor, intra-heterogeneity and inter-heterogeneity. So you could see that when we are growing these cells in the culture, we could see that there are different kind of diameter of the clusters. And this is again showing the heterogeneity of the tumors, with how heterogeneous these tumors are. So based on that, we have classified the clusters, and you could see that based on the DAPI, mean DAPI intensity, we categorize this cluster formation in the microwaves. There are three different kind of clusters: very tight, tight, and loose clusters. So we have seen in multiple samples while we are culturing them. And this is how it looks like. 4 different cancer, breast and lung cancer. Whenever you grow these cells in the microwaves, you see the clusters which shows mostly tight compared to bladder and esophagus cancer. And the part of the reason is that we know that the breast and lung cancer having more number of CTCs getting disseminated. And that is where the more number of CTCs, more the tumor cells, the more tight the clusters. And more tight the clusters, the more metastatic property. So this is something beautifully you could actually see what is happening in ex vivo culture. Now, the clinical perspective, what I mentioned in the beginning, that does it make sense to clinician? And what you can see, the title of the slide, CTC cluster formation. Can it act as a predictor of treatment response or therapeutic resistance? Looking the clusters, can I tell you that if it is, or if it can predict the response or the therapeutic resistance? So this is something, the title of the manuscript, you can see the circulating tumor cell clusters phenotype allows monitoring response to treatment and predict survival. Yes, actually the clusters will allow you to monitoring the response and treatment and predict the survival. And I'll give you some of the examples which are extraordinary examples. So we have collected the samples from the patient, and this is we call longitudinal sample. sample has been collected over the time when the patient has been given treatment. Now, this is an example where the patient has been given gefitinib. Gefitinib is given for the targeted therapy when there is a EGFR mutation, either erlotinib or gefitinib, non-small cell lung cancer. Gefitinib has been given, the patient has been presented in the clinic. After the ninth week, patient were responding well. And at the 12th week, the patient stopped responding to the gefitinib. And for clinician, what they can do, they can just go and do the scan and they will see that, okay, there is no shrinkage of tumor. This is what you can see, the tumor is progressive. And the clinician decided that they need to change the therapy. And they have changed the therapy from gefitinib to pemetrexed and carboplatin 4-cycle. And you could see that the patient responded very well. 9 months, we have collected the sample and the patient is well doing. Now, what you see that to the clusters, if you see, we don't see clusters when the patients were responding. We see clusters only at the 12th week of the therapy, when the patient stopped responding. And again, at the 9th month, the blood sample collected, clusters, you don't find any clusters. This is extraordinary that you could actually replicate and the patient may not need to go every time for the scanning. You can take the blood, you can culture them, and you can see that if the tumor is progressive or non-progressive. And there are many more examples. I will just quickly highlight some of them because I want to stick on time. So this is something the patient has been given for the breast cancer. You could see the CT scan where the tumor progressed after the treatment. And the cluster shows the same, that the 12th week of treatment, you can see the clusters, and at the baseline, there was no clusters. Another example for the triple negative breast cancer, the patient has been given gemcitabine and carboplatin, and there's no progression of cancer. And this is what you can see again, before treatment, after treatment, there's no much changes in the scan. Similarly, the cultures showing the similar kind of examples. This is a unique example where patient has been given immunotherapy. And immunotherapy is something which we consider that it may work, but there are certain group of patients where we have no idea if the patient will respond on the immunotherapy. And that population is something around 10 to 12% of the population. So for the clinician, again, it's a big challenge that should they opt for immunotherapy or not. And this is one of the examples when the immunotherapy has been given and patient has gone for HPD. We call it hyper-progressive disease. And you can see multiple metastases happened. Patient eventually died. So this is again something what you can predict using this liquid biopsy method, where you can see the cluster formation, which could be replicated. You can collect the samples and you can see the response on the clusters. If the patient is responding to the immunotherapy or it may not respond to the immunotherapy. And this is the Kaplan-Meier curve, which shows that the survival of the patient inversely correlate with the cluster formation. The tighter the cluster, the less the survival. So this is again a very good predictor for the disease progression. You can predict the survival. This is a study again in a very large cohort. It's a clinical trial. We were involved in the breast cancer, and patient has been given sunitinib, and then the mastectomy had been done, and we could again predict a similar kind of correlation that the cluster formation in the patient correlate with shorter overall survival. And you could clearly see the Kaplan-Meier curve. So the conclusion is that the cluster formation provide a unique method to predict the treatment response. And the persistence of a cluster would definitely tell you that if it reflects the therapy resistance. I just took one slide to the genomic sides just to make you understand that what I have mentioned so far, it was based on the CTCs. But what you can do with the ctDNA, if you can isolate those DNA from the plasma, if you can do the sequencing, you could actually predict and find the best way of the treatment to the patient. And this is where you can see unique example. The patient has been given initial diagnosis, EGFR mutation was found, gefitinib was given, and gefitinib targeted therapy. 2 cycle, the patient got resistance to the gefitinib. And the second line of therapy for the lung cancer is osimertinib. And you could see that osimertinib has been given to the patient. And again, patient stop responding, and you can see there is a liver metastasis. So no way the clinician can ask the biopsy sample. So they thought, why not they take the blood of the patient and find the mutation. And this is where they took the blood, isolated the plasma, made the sequencing, and they found there's amplification of MET. There's 12 copy number amplification of MET. And the patient has been given, based on that diagnosis, osimertinib and crizotinib. And patient responded well with the treatment. So this is something you could see the impact of the liquid biopsy, how you could rescue, how you could redesign the therapy, and how you could implicate the information, the knowledge you would obtain using genomics. So this is something I was mentioning you, that this is the ultimate goal, that you do different kind of drug susceptibility testing, and you can maneuver different kind of drug to find that what is the best drug work for the patient. And this is where you could see that if it predicts the personalized therapy. So the dream of the personalized therapy, we always call that we need to find the best targeted therapy. If we are not going to find that, the therapy, the effect of the therapy will be cytotoxic. You would find many of the cohort where patients stop responding to the therapy, and this is where we call the non-responders. However, if we could escalate our information and to predict the patient cohort who could be given the best targeted therapy, you would see a different result. And this is what you call the individualized therapy. So I would conclude my talk. I think I'm almost done in 30 minutes time. And this is where I would acknowledge all my collaborators. And thank you very much for your attention, and I'll be happy to take the questions.